brains tend to be smaller, to bolster the view that women are intellectually inferior to men. To date, no one has uncovered any evidence that anatomical disparities might render women incapable of achieving academic distinction in math, physics, or engineering, and the brains of men and women have been shown to be quite clearly similar in many ways. Nevertheless, over the past decade or so, investigators have documented an astonishing array of structural, chemical, and functional variations in the brains of males and females. These inequities are not just interesting idiosyncrasies that might explain why more men than women enjoy the Three Stooges. They raise the possibility that we might need to develop sex-specific treatments for a host of conditions, including depression, addiction, schizophrenia, and post-traumatic stress disorder, PTSD. Furthermore, the differences imply that researchers exploring the structure and function of the brain must take into account the sex of their subjects when analyzing their data and include both women and men in future studies or risk obtaining misleading results. Sculpting the brain. Not so long ago, neuroscientists believed that sex differences in the brain were limited mainly to those regions responsible for mating behavior. In a 1966 Scientific American article entitled Sex Differences in the Brain, Seymour Levine of Stanford University described how sex hormones help to direct divergent reproductive behaviors in rats, with males engaging in mounting and females arching their backs and raising their rumps to attract suitors. Levine mentioned only one brain region in his review, the hypothalamus, a small structure at the base of the brain that is involved in regulating hormone production and controlling basic behaviors such as eating, drinking, and sex. A generation of neuroscientists came to maturity believing that sex differences in the brain referred primarily to mating behaviors, sex hormones, and the hypothalamus. That view, however, has now been knocked aside by a surge of findings that highlight the influence of sex on many areas of cognition and behavior, including memory, emotion, vision, hearing, the processing of faces, and the brain's response to stress hormones. This progress has been accelerated in the past decade by the growing use of sophisticated, non-invasive imaging techniques such as positron emission tomography, PET, and functional magnetic resonance imaging, fMRI, which can peer into the brains of living subjects. These imaging experiments reveal that anatomical variations occur in an assortment of regions throughout the brain. Jill M. Goldstein of Harvard Medical School and her colleagues, for example, used MRI to measure the sizes of many cortical and subcortical areas. Among other things, these investigators found that parts of the frontal cortex, the seat of many higher cognitive functions, are bulkier in women than in men, as are parts of the limbic cortex, which is involved in emotional responses. In men, on the other hand, parts of the parietal cortex, which is involved in space perception, are bigger than in women, as is the amygdala, an almond-shaped structure that responds to emotionally arousing information, to anything that gets the heart pumping and the adrenaline flowing. These size differences, as well as others mentioned throughout the article, are relative. They refer to the overall volume of the structure relative to the overall volume of the brain. Differences in the size of brain structures are generally thought to reflect their relative importance to the animal. For example, primates rely more on vision than olfaction. For rats, the opposite is true. As a result, primate brains maintain proportionately larger regions devoted to vision, and rats devote more space to olfaction. So the existence of widespread anatomical disparities between men and women suggests that sex does influence the way the brain works. Other investigations are finding anatomical sex differences at the cellular level. For example, Sandra